Okay, so I offer today to give a little presentation, so something which would hopefully sort of end our meeting. And I'm at the University of Exeter, as many people know. Officially, I should have retired, but I haven't, in fact. I'm Emeritus Professor of Biological Chemistry there. I'm still officially half-time professor for research only. And so I've carried on maintaining my research, very much involved in biocatalysis. And I thought today it will be good to actually talk to you a little bit about, it's quite a topical area, and I want to try and start with fairly general material. And everybody hears a lot about sustainability. And some of you might have heard about a circular economy. And these are sort of buzzwords which are coming up very much in government plans. And the area I'm involved in biocatalysis, you can see Biocatalysis Centre is based at Exeter University. I had money from the Wellcome Trust for a little building, which is up on the right-hand side of the slide here. So if you come onto the campus, you'll see that building, and that's opposite the Northcote Theatre, so you can't really miss it. And so in there, we work with enzymes and actually using enzymes for various industrial applications is very much in vogue at the moment. It's called biocatalysis or biotransformations. And I want to tell you a little bit about that. Also, I'm very much involved and in fact, vice chair of the European section of applied biocatalysis. They run free webinars. So anybody a little bit more interested in this area, you can join that society for free. And you can have free access to the monthly webinars in various areas of biocatalysis. So some of this work was the other thing on the left-hand side of the screen. The SymBiocat is what's called a cost action. It's where the European Union put together something in a particular area to support meetings, doesn't support actually research itself. And so a lot of this work was associated with something called SysBiocat, which is a cost action. So if I go on, I thought I'd start by looking at these goals. And these are the government sustainable goals. Things which they want to try and put in place by 250, if not before. Many of them here, important goals, all called sustainability. And you can see they cover a very broad area from poverty to zero hunger, good health, well-being, quality education, gender equality, and so on. Many different areas here. And these are all goals which they have, which they want to try and develop. And so many of these actually cut across by definition within biocatalysis. It's very much involved in food production, and I'll give you an example of that. And also there is some involvement with good health and well-being, although it's not listed in the government's. The biocatalysis is very important for drug design, not listed actually in the government list. And certainly affordable and clean energy, and also responsible consumption and production. So it's a sort of very topical area at the moment. So the government goals here, many involve, as I say, the sustainable processes using enzymes. They all tied up with something called a circular economy, which I'll talk a little bit about later. But the goals, if anybody wants to go onto the website address is at the bottom here. You can go and have a look at these DRSM goals and what they define as part of that. So in goal 2, they've got increase in sustainability, productivity, resilience in agricultural, fisheries, food, drink sectors, which biocatalysis is involved in. Certainly ensure access to affordable, reliable, sustaining and modern energy, reducing greenhouse gases. And I'll give you an example where biocatalysis is very much involved in that. And also to ensure sustainable consumption and production patterns, and improve environment with cleaner air, water, minimize waste, and looking at plant, terrestrial and marine wildlife. So biocatalysis cuts across many of those areas. What do we mean by a circular economy? Well, I'm sure a lot of you have heard about circular economy. I don't know, many of you might not have done, so I thought we'd just talk about that. The big thing is, how do we close the circle? So a circular economy, as you can see at the top here, is something where you actually design and you manage, and then any kind of waste is recycled to be used again. So the bottom line is that you don't have any waste. Anything that you produce is recycled and can be used again. And this is what's called the circular economy. So everybody talks about closing the circle, and this is what they mean to this circular type of economy, which is the aim of how we go forward. So I'm using enzymes, enzymes as catalysts, and I'm sure most of you actually know about enzymes, but there's this horrible sort of enzyme metabolic pathway, which I had to learn in my biochemistry lectures. I don't think the students as much learn it off pat as we did, but I used to have to sit and draw out all these intricate pathways where there's many hundreds of enzymes that are responsible for carrying out all the chemistry inside a living cell. And the man standing there is Donald Nicholson. He was very much involved in producing all of these metabolic pathways. You can look his name up, and this is an actual poster of his, which he created. And this is found on the Sigma Aldrich site, if anybody wants to look at that. So it's a very complex pathway of interaction of how all these enzymes control the whole of our metabolism. And they can be actually split up into 6 main groups. This is dependent on which reactions they carry out. And so in a moment, I'll talk about those 6 groups, because some of you might not be aware of that. But one thing about enzymes is that they carry out reactions in a stereospecific way under mild conditions. Well, mild conditions, we don't use heat. Most of them are carrying it out about 30 degrees in our bodies. Under ambient type temperatures. And also the stereospecificity is the handedness. And now, some of you obviously know about chirality, but what do I mean by handedness? Proteins are built up from 21 different amino acids, and in all our proteins, they're in what's called one particular form, the L form. Amino acid has got an amino group, a carboxyl group, a hydrogen group, and then it's got very often which is called an R group, which can vary. And that R group variation determines those different 21 amino acids. The position of that R group can be either on the top, you could imagine, or the bottom. And depending on where that's positioned, then the actual amino acid will have different properties. And so all of the 21 proteins in our bodies are actually optically of one form, and that's called the L form. And that's very important. And that means using an enzyme to do chemistry, which I'll tell you about later, actually means that the product you're going to get is of only one optical form. And that's what drug design people have been wishing for ages, because if you do the drug synthesis using straight chemistry, you usually get a mixture of forms. And if you then use that drug and put it into a human body, then only one of the 2 forms will actually be active and the other one won't, because it's going to bind to an enzyme. And the enzyme is actually going to be specific for only one form. So there's a big advantage in using enzymes because they've got this so-called stereoselectivity. And every time in more complex compounds, every time you have a carbon atom, there is a choice of the arrangement of the groups around that carbon atom. And depending on how the groups are arranged, then that will give some variation to the different forms of the compound, which can actually be present. So it's a very important aspect. So all enzymes carry out reactions in stereospecific ways because there's only one particular so-called substrate or compound which will precisely bind in the active site, which is where the reaction is going to take place. And I'll be showing you some enzymes that I've worked with. So Metabolomics is coming very much into vogue now. A lot of these new techniques are called omics, proteomics, metabolomics. Metabolomics is actually looking at all the different metabolites in a sample. And very often these can be markers of disease. So this has become another sort of important area to look at the particular metabolites. So just to go on to talk a little bit more about what we're talking about before I go on to talking about specific enzymes. These are the different classes, people not aware. There's 6 classes. They're called 1 to 6 here. They all have what's called an EC number. And the easy EC number to remember is alcohol dehydrogenase. This is an enzyme which mops up your alcohol if you've been out drinking the night before. This forms group 1, so it's a so-called oxidoreductase enzyme. And the other numbers, 111, tell you what substrate it uses and what type of dehydrogenases and so on. So if you can't remember any other numbers, if you remember alcohol dehydrogenase 1111, but all of the enzymes on that metabolic pathway I showed you, they've all got individual EC numbers. They will tell you which of those 6 classes the enzymes fall into, what their substrates are, and what other properties they have. So the other thing I just briefly want to mention, because it will come up with some of the enzymes we look at, is coenzymes. Now, it's not all protein. Many enzymes have to have another small chemical compound. These are called coenzymes. They're very often vitamins, and they have to bind in the active site of the enzyme and help with the chemistry that's got to take place. So this is important that we have vitamins in our diet. If we don't have vitamins, we get various disease states. So the enzyme actually has the cofactor bound. When it's bound, it's called an apoenzyme, and without it, it's called a holoenzyme. And sometimes these cofactors stay bound to the enzyme all the time, and other times they have to be recycled. With these oxidoreductase enzymes, like the alcohol dehydrogenase I talked about, they have to be recycled by another enzyme, and that causes some problems in actually using them for biocatalysis. So the cofactor, which the oxidoreductases use, is this niacin, vitamin B3, nicotinamide adenine dinucleotide, NADH+. It picks up a hydride group on this, what's highlighted in red, on the top ring there of the NADH. And you actually can pick it up on one face or the other face of the actual ring there. And there's a related cofactor with just a phosphate group on it, which has got a phosphate group on where the arrow is at the bottom. You can see structurally, it's very similar to the building blocks of nucleic acid. Those of you who are familiar with that, it's got an adenine moiety, a couple of sugars, phosphates, a couple of phosphates, and then we've got this, this nicotinic ring at the top, which picks up the hydride. Enzymes which actually use this cofactor have a very specific structural fold, and I'll talk about some of those later, which actually binds this particular cofactor. So there's many other cofactors, and I won't, or coenzymes as they're often called, but I won't talk about those, but just to mention what they are. So getting back to what I'm sort of interested in, or have been interested in recently, is enzyme discovery. And I've been interested in enzymes which are called thermophilic. The enzymes in our body are only stable to about 37 degrees. If you use those industrially, they're not terribly stable. If you go out and look for in unusual environments, as we've got here, we've got very hot environments. The top one is in a terrestrial environment in Russia. The bottom one is a deep sea geyser, black smoker, the deeper ocean. And there can also be very hot areas of hot springs on other terrestrial sites as well. Yellowstone National Park, for instance, has got a huge number of hot sources. So if you go and look and see what grows there, you see different bacteria and also archaea, and I'll talk about those later. But you can actually look at the genome, that means the genes, and also something called metagenomes. So genomes is actually the gene sequence of some of these novel thermophilic organisms. Metagenome is where you just go and pick up some sample from the environment. It could be from one of these two environments I've got shown here. You don't know what the organisms are there. You just know you've got a bit of DNA. And that DNA can be a mixture from many different things. And it can also represent from organisms which we can't even grow. And as I've put here, we still don't know what the metabolic capability of capacity of life is, and only a small proportion of these catalysts or nature's catalysts or enzymes have been used industrially. We still have new metabolic pathways and activities to discover. And many of these we're finding now in microorganisms that can't be cultured, and also within associated viruses. There is a huge resource of viruses in these environments. And many of them have large genomes. We've worked with one particular marine genome, very big genomes with many different genes on them. So to my surprise, when I started to work in this area, there's still only a smaller number of organisms which can actually cultivate. There's 1 to 2 here, but I think it's about 5% now of known organisms we can actually grow. So if we can't grow them, we don't have their gene sequence, but we can pick them up by looking at a metagenome. So the metagenome is just the DNA in a sample in one of these hot environments, which you can actually pick up. You can sequence the DNA in that metagenome. If it's primarily of one organism which has already been identified, you could say that's what it is. But otherwise, it's a representation of all of this biodiversity which is out there of organisms which we can't grow. So many of the enzymes I've looked at recently came from various projects we've had. One we had was HotZyme, which was a framework EU project. We've had other projects funded by ERA-IB, which is European, but it's funded by the local agency BBSRC here. So many of these projects or larger projects we've had have been characterizing some of these enzymes. So for those of you who are not sort of so familiar here, I've stuck this slide in here, what we mean by bacteria and archaea. So we've got the eukaryotes over on the right side, which we belong to. We have the bacteria over on the left-hand side, as I'm looking at the screen, and some of which are very deep branching. That means they're close to the origin. And then we have this third class, archaea, and they actually branch from the eukaryotic line. And many of these are actually found in these hot environments, and they're what's called extremophiles or extreme enzymes. They grow at very high temperatures, very acidic pH, very high salt, very extreme conditions. And the enzymes which they have are much more robust than the normal enzymes which we would take out of other organisms. So a lot of the enzymes I'll be talking about are either bacteria or archaeal enzymes, and this is where they fit in with the tree of life. The archaea have features which are quite eukaryotic-like. All of their DNA machinery is similar to our DNA machinery, whereas the bacteria are very different. It's interesting to look at, see the evolutionary links to these particular organisms. So back to biocatalysis. We've all used enzymes every day for our everyday life. They carry out important reactions in biosynthesis of safer drug molecules, and I'll give you some examples of that. They can be used, or they are being used a lot in recycling of waste materials, to make this circular economy, to make new chemicals, new biomaterials. Just been looking at some grants today, looking at new biomaterials. And all of this contributes to the so-called circular economy, where effectively nothing goes to waste. We worked a lot with enzymes for the healthcare and detergent industry, cosmetic and food industries, for me, mainly detergent, and also enzymes for carbon dioxide capture. So even for climate change, some very stable enzymes can actually be immobilized onto the various reactor, where they can be put on to the top of chimneys, and they can capture the carbon dioxide, industrially produced carbon dioxide, and they can capture that carbon dioxide, and then it can actually be utilized for other purposes. So they represent sustainable processes, so they tick the sustainability box, which I've explained to you. They're so-called environmentally friendly. That means that the waste products that they produce are not anything like the toxic chemicals, which could be produced chemically. They don't require very high temperatures. So energy-wise, they're much more efficient because they work at ambient temperatures. So generally speaking, they're much safer. They reduce the toxic waste, which is released into the environment. And also the other big thing is that they reduce the petroleum-based products, because many of the products we use today are based on petroleum. There's only an infinite supply of that, and obviously we try to move away from petroleum-based products and make products which we can actually recycle and use again to complete the circle. So what I do in the Biocatalysis Center is a lot recently is discovery and applications of these new enzymes for new drugs. This is one of the examples where we go to one of these hot environments, we isolate the DNA, we can then work out the protein sequence. This is the one-letter code for a protein sequence. Proteins always start with methionine, so it begins with M. And then from that protein sequence, we can actually clone and overexpress the proteins, purify them, crystallize them, the crystallization robot. And then we can actually examine those crystals and take them to what's called a synchrotron to try and work out what their structure is after we've managed to crystallize them. And some of you who have been past Reading, it's not very far from us really, the Reading, one of the world's sort of best synchrotrons, the diamond synchrotron, is actually based at Reading. And this is a large source of very strong radiation, which can actually be tapped to the wavelength roughly of X-rays. So rather than collecting diffraction data from crystals actually in-house, which we used to do, we go and collect the data much more rapidly at the diamond synchrotron. We can then actually work back to what the crystal structure of the enzyme is. And I'll be showing you some of these cartoon type representations of some of the enzymes. You can see little helices there and arrows. There's two secondary structure elements in proteins. One is called an alpha helix, one is called a beta sheet. And so this particular enzyme here, which is of interest to drug companies, called epoxide hydrolase, has got alpha helix beta sheet. It's called a cartoon representation. It isn't just a pretty picture which is drawn, it's actually drawn based on the three-dimensional structure and the actual position of every atom in that structure in 3 dimensions. But it just makes it easier to represent the structure in this form. So in this way, we can actually work out where the reaction takes place, the active site. And then this was a particular interesting, more complicated chemical resolution, which people wanted to do. So normally with the small molecules, it's called S and R rather than L and D with proteins, but you can see you've got several carbon centers in there. And you can get lots of variations of that particular molecule, depending on where the actual carbon atoms, which they bond to. So this is complicated, it's 1s2r 4r or 1r 2s 4r. So chemical companies want to resolve those. And so we actually use the enzymes, in this case, an epoxide enzyme hydrolase, to do that. So they have building blocks for new drugs. So this is the kind of work I'm going to give you some examples from. So why use thermophilic biocatalysts? I've explained that the normal enzymes in our bodies wouldn't be stable enough to do this. You can actually find the enzyme you want. You can actually interrogate DNA databases that I explained from either genomes or metagenomes to actually identify enzymes. You can actually clone them and overexpress them in E. coli. They're easier to purify because you can just heat up the actual cells. And once you've broken them from E. coli, and only the proteins which are stable will remain. You can actually carry out your reactions you want to do at higher temperatures. And I'll give you some examples of those. And very often they're not the natural substrates of the enzyme. There are substrates which are not as soluble. And so if you can work at higher temperatures, that's advantageous. And they're generally more robust to other additional things like surfactants and other organic solvents which are added in the industrial process. They don't break down so easily by being broken up by proteolytic enzymes. And we've spent a long time looking at the small modifications what the enzymes actually have. If they're stable, you can get one particular important metabolic enzyme from different species. The ones in our bodies would only be stable to 37 degrees. You'd have a homologue of that enzyme in one of these thermophilic organisms, which might be stable to 80 degrees. So how are they stabilized? And it's a combination of very small modifications, which we can identify by looking at the three-dimensional structures of a lot of these enzymes. So some examples. Some are already in actual processes for new drug building blocks. And as I said, many of the enzymes are not used with a normal substrate, but with non-natural substrate. And this was a project which people were looking for enzymes which were able to use a particular non-natural substrate to produce new antiviral compounds, which is important nowadays for carbocyclic nucleotides. And some of these were anti-HIV drugs, and they actually inhibit one of the key enzymes in the virus. So you could either go and just search, you could take various genomes, you could cut the DNA up in little pieces, you could put it into something which will make more of that, and actually you can put it into bacteria, and then you can actually screen the plates or Petri dishes, which you have got the compound which of interest in, the substrate of interest, and then you can see which of the organisms grow on that. If they grow on that with little bits of DNA from a so-called expression library, then you get a red coloration in this case. And this means that you're picking up the amino acid, which can be detected in the reaction with something called an anhydride, which many of you will be familiar with. So in this way, you could actually pick up different enzymes which might turn over this non-natural substrate, which is actually the case. And this was for this antiviral drug. This was done with a company called Keratinase. And in this case, you could pick up different organisms. One was a Coenzymes bacterial species. One was an Archaea sulfolobus species. And these particular enzymes could either take what's called a racemic gamma-lactam, which is a mixture of two optical forms. They'd only work on one, and they would actually leave the one they didn't work on. And so that would be pure. And then that could be incorporated into these anti-HIV drugs. So this is an anti-HIV drug, which is called Zygen, is its commercial name. And the ribose sugar has got a methylene bridge rather than a normal oxygen, and these are called carbocyclic nucleotides. And these are all inhibitors of an enzyme called reverse transcriptase, which some of you might be familiar with. Many of the viruses we're dealing with today are RNA viruses, and they have to have an enzyme to convert their RNA into DNA. So the DNA can then be form double-stranded DNA, which can be integrated into the genome. And this is the case actually for the HIV virus. So this went into an HIV inhibitor, which was similar to AZT, if any of you are familiar with that. The COVID virus doesn't actually have this. It actually amplifies RNA. So I think the drugs reverse transcriptase are not actually working with that virus, but many of the viruses, these kind of drugs are antiviral. So they stop straight away the RNA being made to DNA, so the virus is inactivated. So enzymes can be used for their natural activity, but nowadays many of them optimize for industrial processes, and they can be mutated. And the two main ways which our mutations occur, you can either work out the three-dimensional structure of these particular enzymes by crystallizing them, as I explained. And this was the case with one thermophilic esterase enzyme. You can look in detail at the amino acids which are in the active site and carry out the reaction. You can actually change those to change the substrate specificity, to maybe open up the binding pocket so larger substrates combine, which was the case in this particular esterase. Or you can use what's called directed evolution. Here you don't need to know the structure. This is what Frances Arnold got her Nobel Prize for in chemistry in 2018, which many of you must, I'm sure, know. And really what you're doing in a way is mimicking natural evolution by forcing enzymes to actually be able to work under the conditions you want. You might want them to work under higher temperatures. You might want them to work under and high solvent concentrations. You might want them to work with different substrates. And by going through this particular directed evolution, you can actually form mutations and select for the enzymes which are being able to mutate to actually use that particular substrate or work at higher temperatures or whatever you want to do. So she really proved that people could actually not just take the normal enzymes, that they could tailor-make these particular enzymes for optimizing them for different processes. So this is a very powerful technique. It relies, though, you have a lot of screening to do to actually screen for the enzymes which had the desired properties. Nowadays, really, the two techniques go hand in hand very often. You might take a rational design and then maybe do some directed evolution as well. So we don't just take the enzymes as they are, we can actually optimize them for the industrial process. So a couple example of that, the Green Chemistry Award in 2010 was awarded to two companies, Merck and Codexis. Merck is a big chemical company, pharmaceutical company. Codexis is a smaller company, which is the evolution of enzymes. They decided to get together and find a so-called greener route, that's a non-chemical route, for the manufacture of this particular drug, which is sitagliptin. It's already active drug for type 2 diabetes. So it has a big market. They had tried to look for enzymes. They knew that many enzymes have got a chiral or a specific chiral amine group, and this particular enzyme called transaminase actually can actually put that amine group into the molecule. They had one particular enzyme which they had a slight activity towards one of these non-natural substrates that they were looking at, which was related to the drug, the sitagliptin ketone. And then they evolved it by directed evolution to actually replace this very tedious hydrogenation route which involve many chemical steps. And so huge improvement, 25,000-fold, no detectable of the other enantiomer, which was not active, and also huge benefits in production. So they were some of the first to actually do this green chemistry. Many companies are involved in green chemistry now, and once you can find the correct enzyme and develop it, it's much better than the traditional chemistry route. It's much more sustainable, much more environmentally friendly. So many companies, many new drugs are actually produced with a transaminase enzyme. And so we've worked a lot with these enzymes. They're from thermophilic organisms, many of them, of the just 6 different classes, because some can transfer to sugars, and some can transfer to different amino acids and different properties. So the thermophilic one, which the only one I've put in here, you can see this is the crystal structures of these enzymes, with the helices and sheets we talked about. This one acts as a dimer, so it's got 2 polypeptide chains coming together. It's got 2 so-called active sites where the chemistry is going on. And to actually locate the active site, we normally add an inhibitor to the crystal, otherwise it would turn over the substrate. And in this case, it was something called gabapentin, which partially undergone the reaction and then got trapped. So it located the active site. This requires another type of cofactor called pyridoxal phosphate. The actual thermophilic version of this is actually this hexamer. And this is one way which I don't have time to talk about today, but where enzymes can have higher stability by actually making higher organization states. So these are very interesting, some people think about protein structure as art because they are very beautiful symmetrical type structures. But very much, this is all based on the exact amino acid of every position, 3 dimensions in the crystal in the actual protein structure. So I also talked about just giving you some examples of grants I'm involved in. This is another one which is more for breakdown of waste products. And in this case, it's something called ThermoK. And we produce huge amounts of feather waste annually. And people don't know really quite what to do with it. It's very much under resort and under sort of used. It's now known that it can be broken down to produce high value peptides for food, cosmetics, and also lower for feed fish or organic fertilizer. So this is closing this circle I talked about. At the moment, much of it's discarded, it's dumped or it's burnt, and it's only 1/3 recycled, but it's recycled by not a very good method because it's mechanical disruption and high heat and chemicals, which are things we want to try and avoid. And so we're going to use enzymes to do that. And I have a colleague in Bergen who actually is working with this bacteria called a Pyridobacterium species. And you can actually put feathers into this culture of this bacteria, and you can actually watch them degrade, which is quite interesting. So the enzymes which do that are of particular interest. And we look at other enzymes as well, which are being known to have what's called keratinase activity, because the feathers are made up of a structural protein called keratin. And you have to actually break that. There's a lot of disulfide bonds, which are covalent linkages in the protein structures. And we are looking at mixtures of enzymes and interrogating some of the thermophilic genomic and metagenomic databases we have at Exeter, in the hope that we can use a cocktail of these enzymes to hydrolyze the feathers so they're not put to waste. This is actually funded by DEFRA in the UK. This is the first time I've had DEFRA funding. And it's by something called FOSS, which is looking at food security and involves also countries it's actually in South Africa and Kenya, the one we're working with. So it's a sort of broad approach to try and look at these kind of waste products to maintain the circular economy and get value out of them. So you're actually, rather than throwing them away or dumping them, you're actually getting value, and you're replacing petroleum-based materials, which is what we want to do. Another one more recently is to make new nutraceuticals. This is, we all know about diabetes, obesity, hyperlipidemia, hypertension. We all got these, hope we haven't got these diseases, but I think in the Western world, with the intake of sugar and fatty foods, certainly we need to look at this. And some of these rare sugars can be important building blocks. I mean, this is D-xylose, which is a precursor for many drugs. And each also treating, the drugs can treat murine cancers, but also it can be used for nutraceuticals. And we've actually recently got this very thermostable enzyme, the most thermostable sugar isomerase known, from a Thermophilium species. And it's active above 95 degrees centigrade. So really, really high temperatures, amazing. So, and again, we've looked at the structure of that. It's a dimeric enzyme. It's held together with a disulfide bond, which is when two cysteine residues come together to form the only covalent bond in these three-dimensional structures. All the other bonds are fairly weak bonds. And it also, we can work out the location of the active site with a sugar, which is not turned over very rapidly. This particular enzyme is very specific for the luxose sugar, whereas other forms of it are not so specific, and they can use the mannose sugar. So you can use mannose as an inhibitor to trap it in the active site. You can also work out the mechanism of the enzyme. So you're knowing a lot more enzymology, but that actual information is quite important in their actual application industrially. I work a lot with Unilever, as I explained. This was something, the BBSRC, I think this was a blog or something on one of the social media sites, which they highlighted the work we were doing with Unilever. And we've been looking at new enzymes, some which work at lower temperatures, because if you can do your wash at 20 degrees, you use 70% less electricity. Unilever are a company who are very much involved in sustainability. Also, there are stains on your clothes from sebum, or sweat is the, it's not taken off, you get these dark marks, which you can't get rid of. And we've had Innovate UK funding to look at novel enzymes which break down components of sebum, which is very complicated actually, because going to Unilever, we were looking at the different components in the NMR, and you can just take, not even when you're hot, you just put a cover slip on your forehead and then wash whatever's on it with acetone or some kind of solvent. You can analyze in the NMR and see all the different components. And so all members of our team had this particular test done on them. And I thought it would be different, but there's no difference between men and women. So, and you could actually see that it was very similar to all members of the team of these components. So some of them are things like cholesterol, obviously, and some of them are things like squalene, which are quite difficult to break down. So there's a lot of other enzymes there to look and add for those particular applications. Finally, I was going to talk about the carbon dioxide capture, because this again fits very much with the climate side of things. We worked with this particular thermophilic enzyme. It came from a bacterial species called Thermovibrio aminophorecans, and it's interesting. It was isolated from deep sea hydrophobic vent that I showed. Not that particular one, but at depths, the same kind of vent below the sea. And it's interesting because it actually is an anaerobe, doesn't need oxygen, And it actually grows on the presence of carbon dioxide and hydrogen, and it reduces nitrate or sulfur to ammonia and hydrogen sulfide. So one thought that it must have a pretty good carbonic anhydrase enzyme, and it had one of this alpha type of carbonic anhydrase, which is similar to the one that we have, which very much picks up carbon dioxide, and it forms the carbonate. The direction of the enzyme is very much in the forward direction to pick up the carbon dioxide. And we were able to work with Statoil on this particular project and develop this, well, we not develop it with them, but to actually discover this particular enzyme. We did a crystal structure of it. It contains zinc, metal, but also the thermophilic one is a higher molecular weight. It is 4 polypeptide chains joined together, and they're actually joined with disulfides, which are protected against breakdown. In the cell, people think it's a reducing environment and they wouldn't be able to survive, but nature seems to have got a way of actually protecting these disulfides, so they're not available to actually be reduced. And in this case, there's another amino acid which lines they're called lysine residues, basic amino acids, which actually protect the disulfide bond from breaking. This has now been picked up by a company called CO2 it's called Solutions. I was thinking of the other second word. And this is in Canada. I actually did the structure of this enzyme, and we had it with Statoil before they were using it. But Statoil decided that they weren't interested in carbon dioxide capture any longer. They used to pump it down below the seabed. So they then decided that they didn't want to patent the enzyme, but then this other company patented it before us, and they're now working, started to produce these commercial CO capture units where they can actually capture the CO2, and then CO2 can actually be used for other purposes. So I think we missed a trick there. So we should have be doing something. And now the UK are getting interested in this kind of carbon dioxide capture as well. So I had a lot of members involved in some of these grants and funding from quite a few companies. Sigma Aldridge, Novozymes with the Hotzyme grant, Statoil, the one I talked about, GSK, that was Unilever, and recently Evonik. I actually found the new logo here for DEFRA. So we're now, after Brexit, the logo seems to have changed. So we've got a Union Jack on it. So they helped to fund a lot of the work. And I was just going to briefly tell people about what goes on in Exeter, really, is sustainability. There's many institutes now at Exeter University. There's the Sustainability Institute, which is in Cornwall. Perhaps we should get some of these people to talk on some of our lectures. There's a Global Systems Institute. There's something called Eugenus, which is looking for the philosophical and social applications, and also My Biocatalysis Center. And I left this slide because it was where I was showing it to people who didn't know where Devon and Cornwall were, but you can see we cover quite a large area there. And that the Sustainability Institute was actually funded by the EU before Brexit, obviously, because Cornwall was considered a deprived area. So this is on the Exeter Cornwall campus in Penryn in Cornwall. So we might want to go and visit this sometime. They do many master's programs all involved in environmental, conservation, sustainability areas, if people are interested. We've even got an institute for circular. This is actually the business school. We've got a centre for circular economy. So Exeter are taking the circular economy very much in with business, and I think which it needs to be, really, because it isn't just one discipline. They're all very much multidisciplinary areas to address these challenges. And so there's an Exeter MBA which is fairly high ranked. And so we get many international students going on these courses. And then very finally, we all know that the head office for the Met Office is in Exeter. And there's also an Exeter Institute, which is associated with that. So I think a very important area and a lot to offer in the future.